Hello students, welcome to Trunkie Academy. In the past few classes we studied about the biotechnology. We studied about the biotechnological processes, the principles involved in the biotechnology, then applications of biotechnology in agriculture and medicine. Okay. Now, after that, there is one important topic that we need to know with clarity, okay, which forms the basis for the important molecular biology, that is the structure of DNA. Okay, this DNA is such a unique and important structure, the substance or chemical substance that plays a very important role in uh, you can say the transmission of characters from one generation to another, and also um, you can say the regulating the other different functions of the body. Okay, when we say DNA, it is a genetic material. Okay, so many different scientists they worked on the structure and the discovery of DNA. They were trying to understand the which is the genetic material. Is it the DNA or is it the RNA or some other substance which acts as the genetic material? You know that the genetics is the study of the transmission of the characters from parents to offspring, from one generation to another, and the heredity is the process, okay, a procedure, a method by which transmission of characters from parents to offspring takes place. Okay, so the full form of DNA is the deoxyribonucleic acid. When you study about when you study in the uh, nucleotides or nucleosides in class 11, you must have understood that how this DNA is made up of. Okay, there is a nitrogenous base and there is a phosphate group and a sugar group. Okay, sugar molecule, right, joined, and this form the nucleotide. And DNA is the deoxyribonucleic acid. Okay, so DNA as an acidic substance present in the nucleus was first identified by Frederick Misser in 1869. He was the scientist who first identified that this is an acidic substance is present in the nucleus in the year 1869. Okay, he named it nuclein because that time he didn't know that this is a DNA. And however, due to technical limitation in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive and for a long period of time. So it was not clear because see, DNA is a very long polymer, okay, and this such a long polymer, when the technology is not so advanced, okay, and to elucidate and to understand the structure completely, it was not so easy at the time because the technology was not so advanced, and it remained elusive for a long period of time. Okay, but when it was in only 1953 when James Watson and Francis Crick, based on the X-ray diffraction. Now this X-ray diffraction was developed by that time in 1953, and with the help of this technique, James Watson and Francis Crick, they data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. Before that, some other scientists they also tried to describe the structure of the DNA, but that was not satisfactory and was not acceptable by other scientists and other scientists who researched on this structure. Rosalind Franklin, a student of Maurice H. F. Wilkins at King's College in London, took X-ray diffraction pictures of crystalline DNA. Okay, while studying about the structure of DNA, Rosalind Franklin, she was the student of Maurice Wilkins, okay, at King's College in London, and she took the X-ray diffraction picture of crystalline DNA. So this X-ray diffraction picture. Okay, this is the one. Okay. One of the hallmark of Watson and Crick proposition was base pairing between the two strands of polynucleotide chain. However, this proposition was also based on the observation of Erwin Schagger. Now you understand this. One of the important and the unique and the hallmark of the DNA model, okay, of the description of the DNA model given by the Watson and Crick, okay, proposed by the Watson and Crick, the most important point to be noted was, and noticeable, that was the base pairing between the two strands, okay, the hydrogen bonding. But this was also noticed by the Orvin Shargaff, that for a double-stranded DNA, the ratio between adenine and thymine, the base pair, the adenine and thymine, and that between the guanine and cytosine are constant and each equals 1. So if there is a DNA, there are 59 adenine, then that must be the ratio, okay, ratio between the adenine and thymine, if it is 1:3 and 1:2, and then guanine and cytosine is 1:2 or 2:1, it will be constant, okay, it will be always good, okay. So that is the reason. That is called the along with Wilkins, Watson and Crick were awarded Nobel Prize in 1962. So along with the Wilkins, Watson and Crick were awarded Nobel Prize for discovery of the structure of DNA. Okay, so these are the pictures of those three scientists who were involved in the discovery of or understanding of the or proposing the structure of DNA. Okay, it has double-stranded, okay, DNA structure of the DNA. Okay, so Francis Harry, Crompton Crick, James D.V. Watson, and Maurice Hugh Frederick Wilkins. Okay, now the base pairing confers a unique property to polynucleotide chains. Okay, so when there is a two strand in a DNA, these strands are joined end to end, joined by hydrogen bonding. Okay, so this pairing, one base and another base, one base in one strand and another base in another strand. So two strands, and they are joined by the hydrogen bonding. So this base pairing confers a unique property of polynucleotide, because single strand DNA, okay, is joins with another strand to form double-stranded DNA, and this double-stranded structure of the DNA confers a unique property for this DNA or polypeptide chain. The chains are complementary to each other, and therefore, if the sequence of bases in one strand is known, then the sequence of other can be predicted. Because if there is adenine, then there will be thymine. So it is always complementary. The strand of one strand of DNA and the base pairs which are present in the one strand, that base pair will be complementary to the another strand, so that will be joined with another strand by complementary bases. Okay, so if you know the bases in the one strand, then definitely will be able to know the bases is another strand that will be easily predicted. Okay, because they are complementary. Also, if each strand from DNA, let us call it parental DNA, acts as a template for synthesis of a new strand, the two double-stranded DNA, and let us call them the daughter DNA, thus produced would be identical to the parental DNA. Understood? Now means if each strand of DNA, suppose that is the main which is going to replicate, okay, acts as a template for synthesis of new strand, the two double-strand DNA will be formed in the daughter strand. Now the new produced would be identical to the parental DNA. Because of this, genetic implications of the structure of DNA became very clear due to this feature of the, okay, because the DNA, a parent which is replicating, okay, forming acts as template for the synthesis of new strand, or it's not replicating, or if, for example, if we are taking one DNA, single strand, and this is forming a template for the new double strand, so that will be identical to the parent DNA molecule. Okay, because of this, genetic implications of the structure of DNA became very clear. Okay, so structure of DNA. James Watson and Francis Crick proposed that DNA consists of two strands which are helically coiled. Okay, so that you have already seen in many pictures. It is helically coiled and they run anti-parallel. Okay, now you see. So double helix is right-handed and is referred to as the beta form or B DNA and most common model. Okay, so there are other model also. The most common is the B form or B DNA, which are coiled helically and they run anti-parallelly. Okay, DNA is formed of millions of deoxyribonucleotides. That's why it's called deoxyribonucleic acid, one is joined with the another deoxyribonucleic acid. That is why it's called the polymer. Polymer. Okay. So it's a long polymeric chain of deoxyribonucleic acid. Nucleic. Okay. Many of the salient feature of double helix structure of DNA are as follows. It is made up of two polynucleotide chains. I told you, in one polynucleotide, polymer of nucleotide chain, okay, and where the backbone is constituted by sugar phosphate and the base project inside. Okay, so you can see that the two chains have anti-parallel polarity. This means that if one chain has polarity 5' to 3', then other has 3' to 5', just opposite in direction. The bases in two strands are paired through hydrogen bonds, okay, forming base pairs. Adenine forms two hydrogen bonds with thymine and thymine from the opposite strand and vice versa. That's why adenine is joined with the thymine from one strand. If the one strand has the thymine, the other will have adenine, and they are joined by the two hydrogen bond, and vice versa. Now similarly, guanine will be bonded with the cytosine with 3 hydrogen bonds. Okay, so as a result, a purine always comes opposite to a pyrimidine. So there are two types of nucleotide we know, purine and pyrimidine. Okay, so purine will always come opposite to pyrimidine. Now this generates approximately uniform distance, 20 angstrom between the two strands of the helix. The two chains are coiled in a right-handed fashion. Okay, right-handed fashion. The two chains are coiled in right-handed fashion, and they have got distance, uniform distance of 20 angstrom between the two strands. The distance. Okay, if you say talk about the distance, okay, so there is the distance of 20 angstrom. The pitch of the helix of the turning one coil, okay, is a 3.4 nanometer, is 1/1000th of a meter. That is a 10 to the power minus 9 meter, and there are roughly 10 base pair in each turn. So as I told you, in a turn, so in this turn, okay, so this in one turn they will have this turn it will be 3.4 nanometer, and that will consist of 10 base pair in each turn. Okay, so consequently the distance between two successive base pair in a helix is approximately equal to 0.34 meter. So 10 base pair means that will be, so distance between with the successive one base pair to another base pair, then so that then there will be the, since there are 10, okay, so one base pair, two base pair, three pair, so the distance between these one base pair to another, okay, then it will be this 0.34 nanometer. Okay, the plane of one base pair stacks over the other in a double helix. This, in addition to H bond, confers stability of the helical structure. Now you can see here, this is the 1 nanometer distance has been shown here, and this is the 3.4 nanometer here, one turn. See, up to here, okay, so this is the 3.0, and in one turn, so from here it's going to, from here it's starting, and then it's going to have one turn here up to here. So in one turn it is having a distance of 3.4 nanometer, and the 10 base pairs are present in these 3.4 nanometer. Okay, so that means distance between the two base pairs will have this 0.3 nanometer of distance. Okay, nanometer, n, right? Okay, you now see this diagram also, you can see. So 0.3, the one distance here, this one, distance between the one base pair to the next base pair, 0.3, and from here one turn is taking, going, and then coming it here. So here, so from here to here it is the 3.4 nanometer. So okay, so there is the major groove and minor groove are formed thus. So this is the major groove, the bigger one groove, and the minor groove here. Okay, so this turning and coiling is forming minor and major groove. Okay, so this is how the structure of DNA looks like. If two purines form base pair being heterocyclic, they make DNA bulged, or in or if two pyrimidine form as a base pair being monocyclic, the make DNA narrower so that purine always pairs with a pyrimidine and vice versa. Therefore, the diameter of DNA is constant. That is the reason. So pyrimidine will always be pairing with the purine, and that is the reason you can see the purine makes a base pair being heterocyclic. They make DNA bulged. Since it's a heterocyclic, purine is heterocyclic. Purine from the base pair being heterocyclic. So, and if the two pyrimidine form the base, because they are being monocyclic, makes the DNA narrower. So, there is the what we call that, the diameter is always constant. That is the reason. Okay. A specific purine pairs with a specific pyrimidine because of a perfect match between hydrogen donor and acceptor site on two bases. A specific purine only matches or will bind or will be bond with, so hydrogen bond with a specific pyrimidine. Okay, a specific purine will bind with a specific pyrimidine because there is a perfect match between the hydrogen donor and acceptor site on these two bases, specific two bases. Okay. The proposition of a double helix structure for DNA and its simplicity in explaining the genetic implication became revolutionary. When this double helix structure of DNA was proposed, okay, and when the research started about the heredity and the role of DNA in heredity, okay, there was a revolution, and there was a kind of a new era started in the biology or in genetics and heredity. Okay, then was very soon Francis Crick proposed central dogma in molecular biology, which states that genetic information flows from DNA to RNA and RNA to protein. DNA, they are transcribing the mRNA. The message that is contained in the DNA is transcribed to RNA, and this the message contain and then copied or brought from the DNA in the RNA, that is translated into amino acid or protein. So this is called the central dogma of molecular biology. So that means the Francis Crick proposed where the genetic information flows from DNA to RNA and RNA to protein. So replication of DNA, it gives rise to mRNA by transcription. Then this mRNA undergoes the translation to form protein. So this is called the central dogma of molecular biology. In retroviruses, RNA acts as the genetic material. Okay. So as I told you earlier, genetics is the main genetic. DNA is the main genetic material in many organisms, almost every, but few organisms like virus. Okay, it is the RNA acting as the genetic material. Examples: in HIV, the flow of information is in reverse direction, that is RNA to DNA. So it is reverse direction. So it's called, that is why it's called retrovirus. Retro, opposite, back. Okay, retrovirus. Okay, in HIV virus, retro. Now we go through some questions. Okay. Double helix is the right-handed is it is a D form or B form or Z form or X form. It is the B form, right-handed. It's the most commonly found. DNA is formed by millions of nucleotides, nucleosides, or ribonucleotides or deoxynucleotides. It is the deoxyribonucleotides. Okay, it is the polymerase. Okay, it's a polymer, so not polymerase, it is a polymer, okay, or polymer, a large or long polymer of the deoxyribonucleotide. A DNA duplex has two strands with a diameter of 10 angstrom or 20 angstrom or 3 angstrom. It is the 20 angstrom. So distance between the strands, okay, in the duplex, okay, that is the 20 angstrom. Okay. Now we'll come to another important topic, that is the packaging of DNA. Okay. While discussing about the prokaryotic and the eukaryotic cells, I also told you about that eukaryotic cells are much more advanced in the respect or that they have got membrane-bound cell organelle. Okay. They have got a well-defined nucleus and prominent nucleus present in the cytoplasm in the central region of the cell. And the chromosomes are are well packed, the DNA are well packed, and they are in the nucleus, and the nucleus has well defined envelope, nuclear membrane, and the nucleus is prominent and distinct. Okay, that is the difference between prokaryotic and eukaryotic. So when we talk about the well packaged or DNA is well packed, that means it has got important kind of structure which is not found in the prokaryotic cells. Okay, that is the reason the genetic material is called that. Genetic material is naked in prokaryotic cell. Okay, so packaging of DNA helix taking the distance between the two consecutive base pairs, that is a 0.3 nanometer that we studied just now. Okay, so the distance of one turn, okay, one turn is having the 3.4 nanometer, and each distance between the each base pair is, then it will be 0.3 nanometer. Okay, the length of the DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of base pairs with distance between the two consecutive base pairs, that is 6.6 into 10 to the power 9 base pair into 0.3 nanometer into 10 to the power minus 9 meter per base pair, turns out to be approximately 2.2 meter. Just imagine, so small, microscopic, but so long, 2.2 meters. Okay, so are you getting my point? Or taking the distance between two consecutive and then if it in a typical mammalian cell, if you want to calculate, okay, so the total number of by multiplying the number of base pairs and then multiplying with the distance in the distance between the base pairs, will get about the length of this as 2.2 meters. Just imagine, it's a very, very long, so small, microscopic. Okay, now this length is far greater than the dimension of a typical nucleus. Can you imagine? Cell itself is microscopic, and within the cell is the nucleus, and within the nucleus is this chromosomes, and within the chromosomes is this DNA. So just imagine how small is this, and even though this is a very long, isn't distant. So this length of 2.2 meter is very far greater than dimension of the typical nucleus, approximately 10 raised to the power minus 6. 10 to the power minus 6 meter is the dimension of the nucleus, where we find 2.2 meter long DNA. Just understand. Okay, so how is such a long polymer packaged in a cell? So because this is packaged, this is the magic of packaging. Okay, it's packaging in such a way so it is accommodated in such a small dimension of the nucleus. Okay, so answer to this question can be obtained in this topic that we are going to study, the packaging. The length of eukaryotic DNA is far greater than the dimension of a typical nucleus. Nucleus dimension is about 10 raised to the power minus 6 meter. Okay, so chromosomes, you can see, is highly coiled, and where we find highly coiled DNA. Okay, the DNA of prokaryotes and eukaryotes are packed in a space which is 1,000 to 10,000 times smaller, and such a packing occurs differently. Okay, in both. So packing in prokaryotes such as Escherichia coli, through they do not have a defined nucleus. DNA is scattered throughout this cell. I told you when explaining about the difference between the prokaryotic and eukaryotic cells. So DNA is scattered. Okay, there is no distinct nucleus. Okay, and there is no included in the nuclear envelope, and DNA is dispersed in the cytoplasm. This is a bacteria microplasma. Okay, the prokaryotic cells. Now DNA, being negatively charged, is held with some proteins that have positive charge. Okay, in a region termed as nucleoid. So they, in the cytoplasm of this bacterial cell, that is the prokaryotic cells. Okay, there is some kind of accumulation in the central region, the DNA, because of some proteins are having positive charge. Okay, and the DNA is having some negatively charged, so that interact and they are accumulated somewhere in the central region, and that is termed as nucleoid. Okay, so the DNA in the nucleoid is organized in large loops held by proteins. So these are organized by, held by proteins. They form a large loops and they're held by proteins. The DNA of prokaryotes is almost circular and lacks chromatin organization. Unlike eukaryotic cell, where we find the chromatin fiber, okay, there is no such things in prokaryote. They are circular. DNA is circular, okay. This is referred to as genophore. DNA packing in eukaryotes. In eukaryotes, this organization is much more complex because there is a high coiling, and also there is a very good packaging, okay, very compact packaging, and that makes its structure very its organism very complex. There is a set of positively charged and basic proteins called histones. So 8 proteins octamore, they forms a nucleosome, histones, okay. So 8 H2A, H2B, H4, H3, these are the core of 8 histone protein, and on which this the DNA is wounded, forming one unit that is called nucleosome. So a unit of this 8 octamore protein, histone protein, this is called nucleosome, okay, nucleosome. And each nucleosomes are again they are adjacent to each other and they form tightly packed, and then it's present in the chromatin fiber, then in the chromosome, okay. So histone protein, okay, 8, there are 8 in one nucleosome. So, okay, so a unit of 8 is called nucleosome, okay. The DNA are wounded over this 8 histone protein. A protein acquires charge depending upon the abundance of amino acid residues with charged side chain, okay. So a protein acquires charge depending on the which amino acids are there in the protein. Abundance, if the amino acids are more in the protein, okay, so depending on that there will be charge in the protein, okay. The primary protein structure, it is the linear structure, primary. There are primary structure of protein, there is secondary structure, tertiary structure, and quaternary structure, okay. If there's simply a polypeptide chain, is primary. If it is the alpha helix, then it is the like find in the keratin, this is the secondary, okay. And if the hemoglobin is the quaternary structure, example, okay. Now histones are rich in basic amino acid residues, lysine and arginine. So accordingly there will be the charge. So these are the basic, okay, basic amino acid residues, and they are both amino acid residue carry positive charge in their side chains, okay, both of them. That means I told you here basic amino acid are rich lysine and arginine, and these lysine and arginine residue carry positive charge on their side chains, okay. So you can see the nucleosomes and how they are again packed to form the chromatin fiber chromosomes, correct. So here, so this one unit is called the nucleosome. So this one unit is the nucleosome, and this nucleosome has 8 octamer of histone protein, right. These are histone protein, they are forming this. Now, so you know, just histone are organized to form a unit of 8 molecules called histone octamer, okay. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. I told you that time, right. So negatively charged DNA, which has got slightly negative charge, is wrapped or wounded around the positively charged histone octamer, and that wounded DNA over the 8 histone octamer protein, that is called the nucleosome, okay, one unit. So here you can see H2A, H2B, H3, H4 and H2B. So 8 histone and the DNA are wounded. The negatively charged DNA wounded over, wrapped over the positively charged histone protein, okay, to form a nucleosome. So this is here how the nucleosome is formed. This nucleosome structure is not found in histone protein, is not found in prokaryotic cell. It is found in the eukaryotic cell. Okay. Now histone protein and the wounding, okay, wrapping of the DNA. So this makes one nucleosome unit. Okay, a typical nucleosome contains 200 base pairs of DNA helix. Okay, a typical nucleosome will have the 200. So while wounding, wrapping around this 8 octamer histone protein, the DNA will have that the DNA present in one unit of nucleosome, there will have 200 base pair of DNA helix. 200 base pair of DNA helix. Okay, nucleosome constitute the repeating unit of structure in nucleus called chromatin. So what will happen? These nucleosomes constitute the repeating unit of structure of the nucleosome, and then in the nucleus and called the chromatin. Okay, nucleosome in chromatin are seen as beads and string when viewed under the electron microscope. Okay, so like I showed you here, this picture you can see here, beads on a string, beads on a string. Okay, then again they are packed to form the chromatin fiber. Okay, so beads, and it looks like a beads and a string. Okay, so when viewed under microscope. Okay, histone octamer, and the 10 nanometer is the size of that octamer histone protein. Okay, the core DNA. Okay, and now you see the beads and string structure in the chromatin is packaged to form chromatin fiber that are further coiled and condensed at metaphase stage of cell division to form chromosomes. So these chromatin fibers are further condensed. Okay, coiled and condensed, again coiled and then condensed, the metaphase stage of the cell division to form the chromosomes. Here, so okay, the packaging of chromatin at higher level requires additional set of protein that collectively referred to as non-histone chromosomal protein, NHC. Okay, so further packaging of chromatin fiber, condensing and coiling. Okay, I mean the forming a compact. Some other proteins are required. Okay, and these are called non-histone chromosomal protein, NHC. In a typical nucleus, some regions of chromatin are loosely packed. Okay, and are referred to as euchromatin. In some part. Okay, in some region of the chromatin, they are loosely packed, and they are called euchromatin. The chromatin that is more densely packed and that takes the stains, dark stain, that takes the large quantity of and that is stained darkly, then it is called heterochromatin. So there are two regions which are, some are loosely packed and one is darkly packed. So darkly packed will have the dark staining and the loosely packed will have the light staining. Okay, so light staining region, that's called the euchromatin, and the darkly packed and darkly staining is called the heterochromatin. So euchromatin is transcriptionally active. Okay, the one which is lightly packed, loosely packed, that is the transcriptionally active, whereas heterochromatin is inactive. Okay, so that is tightly packed, which is inactive form of the. Now we will go through some questions. Histones are rich in basic amino acids like lysine and arginine, alanine, valine, or lysine arginine, or lysine alanine, or alanine lysine. It is the lysine and arginine. Okay, basic amino acids are present. Okay, so that there is the negatively charged and positively charged due to that interaction. The wounding around that histone octamer is possible. A typical nucleosome consists of 100 base pairs of 200 base pairs, 400 or 300. It is a 200 base pairs. Okay, it is a 200 base pairs of DNA helix is there in one nucleosome unit. Okay. Now, so this was all about the DNA structure of the DNA and how this DNA are packaged and present in the nucleus. Okay, this packaging, condensing, coiling is the indication of the advancement of the cell and the nucleus. Okay, and also in the course of evolutions, eukaryotic cells and the nucleus of the eukaryotic cells are actually more advanced than the prokaryotic cells. Okay, and this was all about the structure of the DNA and how this DNA are packed and packaged and present in the nucleus and in the chromosomes. Okay, So we studied about that there is the DNA is the polynucleotide, okay, and the two strands are joined, okay, by hydrogen bonding, and one strand and another strand. Both the strands have the base pairs, but the base pair will be complementary to each other. Okay, if you know the base pair of one strand, we can easily predict the base pairs on the another strand. Okay, and there is a kind of right-handed turning helix that can be formed. Typically, normally this is found in the other form, although Z form and other forms also are there, but B form right-handed coiling is the common one, and when we find the one turn will the length of that will have the 3.4 nanometer, okay, and that consists of 10 base pair, so the distance between that between the each base pair will be 0.3 nanometer, okay. So then we studied about the nucleosome, histone proteins. 8 histone proteins are there forming octamer, and then on which the DNA are wounded, okay, and that forms the beads on a string structure, and this forms the chromatin. These chromatins are further condensed, okay, during the metaphysical state of the cell division, and this condensing, coiling, and compacting requires some additional protein that is called non-histone chromosomal protein, and that helps in the forming the compact and more thick to change to form the chromosome, okay, to change it to chromosome to form as chromosomes, okay. So this was all about the structure of DNA and the packaging of DNA, okay. So in the next class tomorrow we'll study about the next topic, okay. So thank you for joining and listening today, okay. Tomorrow again we'll meet at the same time, okay, with another topic. So till then, goodbye, take care.